midterm. Next, we'll go over some things related to schizophrenia and more genetics and GWAS studies related to schizophrenia, but that material will not be covered on your midterm. The final tutorial midterm office hours. Some of you have already gotten similar material in my course previously. Some of it is new for some of you. Whatever the case is, I'd like you to drop by if you're having issues with the material. And if you don't understand anything, I want you to make sure that you understand it. I'm going to take you through this one paper. And this paper is actually kind of an interesting paper in terms of neurobiology. And I'll be able to do at the end of this course. I'm not going to give you like rubrics on how to actually read a paper. It's my job to show you how to interpret a paper and give you a spin as a neurobiologist. So I happen to know some of the background for this paper in particular, and this is one of those really interesting papers. This made huge news when it first came out. Smarter mice, right? And everyone had little pictures of Pinky and the Brain, and everyone was thinking that, wow, you know, these smarter mice, what does this actually mean? It was an important paper for me too, because I also want neuroscience students to be thinking about this. And I often ask students who randomly drop by the office, I ask my colleagues exactly the same questions, by the way, that I ask my neuroscience students. How would you design a better neuron? How would you make a neuron function better? How would you make a brain better? And that's one of those questions, the types of questions that are really outside the box, that in theory there are answers for. And this was one of those papers that came out a few years back, about a year and a half ago, that actually addressed one of those questions. If you could make a better brain, how would you do it? And the answer was actually really surprising. And I'm going to go through some of the data with you, and I'm going to go through it in sort of a logical way with your help. And in the second half of the course, you're going to help me in a couple of different ways throughout the course. You're actually going to lecture with me on one lecture, and we'll talk about that later on. But for today, we're going to go through whether or not mice actually is a chimeric brain that involves human cells in a mouse background. They didn't use neurons, which again is maybe surprising, maybe not surprising. And maybe one day, you know, an individual might take human neurons and try to transplant them in. I doubt they're going to make the mice any smarter, and I'll take you through some of the rationale for this. I don't know how much of a background all of you have, and I'm not going to ask you to know things like neuroethology, which is really the comparative study of different nervous systems to get an idea of why are our brains better. Our brains are better, by the way, than other species, but why are our brains better? When we do comparative studies, which we call neuroethology, it turns out that our brains aren't bigger. They're not the biggest in the animal kingdom. They don't even have as many convolutions as other animal species. And certainly our brains are not, in theory, any different than those of primates. But our brains function differently. And people have been wondering, in terms of neuroethology, why is that the case? So again, one of the things that I'm really interested in is for you to understand sort of the molecular, cellular, developmental perspectives on how our brain develops. And I know now, you know, I ask my colleagues to make sure that if they're teaching a course, that you get some of the background information on this, and we'll go and visit and revisit these progenitor cells in a moment. And then we're going to talk about these circuits again. And in particular, we're going to talk about that hippocampal circuit that many of you will remember the anatomy for. This is where you're going to help me out a little bit, and then we'll go back and revisit this neural circuit. And you don't build a better brain with neurons. It turns out that you build a better brain using glial cells. And this is sort of that big paper that came out in Cell, stem cell biology, and you can understand the reason why it's in stem cell biology Cell and not the main Cell journal, because they actually injected in progenitor cells. And what they found was actually really surprising. But by taking human glial progenitors within the mice, then one of the things that they saw was these mice actually learn better. They learn faster. By every metric that you could throw at them, the forebrain learning actually was better. Surprising. Neurobiologist, and you're interested in this, what other things would you look at to see if this was a good paper? I haven't taken you through this paper yet, and I'm not asking you to evaluate this paper now because we haven't looked at it. But if this is such a big paper, what would you think would happen after this was published way back in 2013, I believe, or 2012? Big paper, yes. Sure, that's one of the things I always ask for you as neurobiology students to be thinking of. If this is a real a few years ago, were there follow-ups to this paper? And in fact, the only follow-up is done by the same group. And right away you're thinking, well, this is that whole PKM zeta story again, right? Because you only have one group reproducing these results. Yeah. Maybe. And actually, that becomes really controversial as well. And I don't know if you've been watching the recent special on Einstein's brain. Like, he hadn't actually given permission for his brain to be taken. There was a lot of miscommunication, and the scientists who took it actually did a lot of unethical things without the family's permission. The glial cells in the corpus callosum are only now sort of being re-examined, so it's possible. Okay, so I'm going to take you through this paper. How would you build this better brain? So we have things that we think, as far as we know, and I'm not talking about the social aspect of the brain, but in terms of our abilities, in terms of thinking as well as intelligence, we tend to score a lot better than any of the other animal species that are out there. And these unique capabilities of our brains, what's unique about them? Our neurons don't tend to be all that unique. Our neurons have the same sorts of channels that every other neurons have. That's one of the reasons why you study all of those other neuron channels. To the brain, it's not the numbers of neurons. I'm sure that's important to a certain extent, but overall, an average person's brain versus an average primate brain versus an average elephant brain, an average dolphin brain, etc. There really isn't that much that really makes the neurons stand apart. Some have slightly different types of neurons, etc. But if we're taking a look, how do we understand the differences that other really large brain species, which is really what neuroethology is? One of the things that may or may not surprise you, depending on how much of a developmental background you have, and I know that many of you don't, is that the other component, the glial cells, the one that outnumbers the neurons about 10 to 1 in some places, 1,000 to 1 in other places, it's really the glial cells that make us stand apart. For this whole paper, that if you're looking at making a more efficient brain, that perhaps we shouldn't be looking at modifying the neurons, we should be modifying sort of the extra neuronal compartment, the glial cells, the other astrocytic cohort that comes along with us. Our astrocytes, our astroglia, the one that outnumber our neurons by quite a bit, are both morphologically as well as functionally distinct from other mammal species. Again, hopefully it's a review for some of you. It might be brand new for some of you. And in particular, we know that these astroglia are actually quite a bit bigger. And I'm going to show you some of the evidence for this in situ, in the mouse brain in a little bit. They also have a number of different phenotypes or morphologies associated with them. They have this cellular pleomorphism, in other words. They can adapt and they show this adaptation. They are also architecturally much more complex than those in other species. These astrocytes, and this very network, because they're bigger, they have more elaborate processes, and they're involved in calcium signaling. They can spread a calcium signal across a wider area much faster because they form what is known as a syncytium, or a group of cells. Okay, so this syncytial group networking ability might make them unique and separate and distinct from other species. And we know for sure that in the mouse, these are unique properties for human glial cells. So is this really what makes us unique, right? So if it is, great. I'm not so sure, to be honest. I'll take you through some of the data, but is this really what gives us our uniqueness in terms of those things like thinking, learning, adaptability, and other things that seem to be part of our human nature? Do you have any questions on this before we go on? Okay. So these overall differences may help us to understand how these astrocytes help to coordinate as well as modulate neuronal signals. If you have a neuron firing here, it might be able to spread that signal out much faster across the cortex because of the way it's connected than mouse astrocytes, which are not as easily connected. So this also gets us to rethink the way in which our brain works. So if this really is that important, we have up until now really been neurocentric, right? And when I ask grad students, when I ask TAs, and when I even ask my colleagues, you know, how would you build a better brain? Some of them have a really hard time. Some of them try thinking about neurons, but maybe our view of looking at building more efficient models of thinking, plasticity, etc., might be due to a support network due to these astrocytic glia. So this is sort of the basis of this paper. Can we modify using this supporting network that has never been the star of neuroscience ever? Can we actually do this in a rational way and demonstrate that they actually perform better? And this gets into this idea that we can take human cells and we can generate these chimeras. So we have human glial progenitor cells that are being injected into early postnatal mice, and that as they grow over time, functions in the mice become any better. So they wanted to know a couple of things in this paper, and you should, anytime you're doing this right from the get-go, you need to be able to demonstrate a couple of things. If you're putting a glial cell back into a non-native environment, so a glial cell in the human is bigger, it's more elaborate, it has lots of complexity. Now you're putting it into a totally new network, a totally new environment. Does it become human? Does it stay human, or does it function more like a mouse cell? And that's one of the important things in science when you're reading about why they're doing these things. It's not to just do these things to validate it. They need to be able to demonstrate all of the different things that I'm going to show you in the first few slides. And you know, is there going to be any component where these mice function better? The title of the article already gives that point away. They do. They function significantly better. And I'm going to take you through some of the data that represents this. So again, in this 2012-2013 paper by Han et al., when they take these progenitor cells, and these are the types of progenitor cells that they're injecting, these progenitor cells in theory can be different glial cells. And again, these neural-like stem cells, which can become slightly more differentiated, or they can become these lineage-committed progenitors. They're already into being different types of neural-like cells, whether it is the APC type or whether it is the O2A progenitor cells, whatever it is, these can become glial cells. And these glial cells in turn can become oligodendrocytes, astrocytes, etc. And if we start it up far enough, they might even become neurons. These are not the group of cells that this paper is dealing with. We're dealing with cells that will eventually become the glial cells. So we're looking at the glial progenitor cells only, the ones that theoretically can become oligodendrocytes or astrocytes, okay? They're not creating humanized neurons. They're only creating these glial progenitor cells. And one of the ways that they do this is actually kind of neat. It was one of the first demonstrations that they could use magnetic separation techniques where they were labeling the cells. And this is something that I want you to know. How do you know a glial progenitor cell is not an oligocyte, oligodendrocyte, or an astrocyte? All of those stains that I keep showing you on a consistent basis allow you to know that there are certain markers or certain cellular markers, molecular markers that are unique to different populations. So when we see that these cells are staining for a cell surface marker known as A2B5, and it's an A2B5 positive cell, we know that we have a glial cell progenitor. If it also turns out that this is polysialic acid NCAM deficient, there's no PSA NCAM, we know that it can't be a neuron. So in other words, we are dealing with a population that will eventually become these differentiated glial cells. Now, I'm hoping that this is somewhat of a review for you, and I know that some of you who are taking 300 this semester right now, hopefully this is a review. Where do you get these progenitor cells from in the brain? Well, one of the things that they got, and these are from human embryos, is from the subventricular zone. Okay, so they got those progenitor cells from the subventricular zone in the human embryonic fetus, and then they were able to separate them and purify them only to the glial progenitor cells. Okay, any questions on this before we go on? So this is how they validated that we have a pure population of glial progenitor cells. They're from a human source. They're pure. And now we're going to see what happens when we put them into the mouse forebrain. Yep, do you have a question?
Yeah, so they were allowed to use human embryos?
Yes, they were. Yep.
What country was it in?
This is an American study. Yeah. I don't know what the political climate was at the time, but they did have these human embryonic glial progenitors. So again, some of this is what we've looked at previously. So you've isolated through this magnetic separation technique, where you have progenitor cells that you are marking with A2B5. They are not neuronal cells because they are PSA or polysialic acid neural cell adhesion molecule deficient. So they're only glial progenitor cells. You get 100% glial progenitor cells. All the other cells are removed. And then once you have them as a pure population, one of the things that you do is before you implant, because how are you going to know that this comes from a human, right? And so the fact that they've got pure cells, the pure glial cells, I'm hoping that you get that from the A to B. But before they did this implantation, they wanted to be able to track these neurons. And it might seem superfluous to you, but this is the way in which science works. So they took a lentivirus, and then they put in GFP under the control of a cytoviral promoter. Of this lentiviral expression. And they use a lentivirus. As we've talked about before, in neuro, we usually have a couple of choices. We have AAV, we have lentivirus, and then we also have the Synvis virus, and in some cases, herpes simplex. The reason why they decided to use lenti is for precisely the reasons that I pointed out to you before. Lentivirus, the expression goes on for a very, very long period of time. And they were able to detect green fluorescent protein, which is normally not found in the brain, at least 1 year in vivo after implantation. Any questions on this before we go on? Pure population of cells being taken from the subventricular zone in humans, purified to glial progenitors only, transfected or transduced with GFP to be able to mark them, and then they're implanting this in mouse brain. Any questions before we go on? Yes, Anne? Sorry? Where? Oh, okay. So you're just going to have to leave that for the time being. I'm not going to get into all the details here. It's a different isoform of a lentivirus. Yep. Okay. So then they take these different glial progenitors that are now labeled with GFP, they implant them into the forebrain, and what they see is this spreads. And so each of these little dots shows that in the adult, to detect green fluorescence protein. So this is the forebrain, and you can see there's lots of expression throughout the entire forebrain and each of the 3 panels around the structures that you see here. So all of those little dots that you see, these sort of gray hashings, are where they see these green fluorescent protein markers in the adult animal after they had transplanted it at postnatal day 1. Okay. What structure is this, just before I go on? That you see here, very characteristic, classic, rodent. Sorry? This is the hippocampus, and again, you see the hippocampal formation down here. This becomes important in a moment. So this is a human cell. So this is not going into a normal mouse. So one of the things that they had to do was to use a very specific mouse, which was immunodeficient. And hopefully you'll understand why it's immunodeficient. It's a mouse. You're putting human tissue in it. You don't want it to be cross. Deficient mouse is the one that actually did these experiments. The mouse. Yes. The severely immunocompromised, immunodeficient mouse, that's one that you should make sure that you know. And now we know about the RAG2 as well. So these xenografts, these transplanted cells, were then allowed to proceed. And not only can they look at GFP, but they can also identify, because the human nuclear proteins are unique and separate from the mouse nuclear proteins, you can also go back and stain for human nuclear proteins. They came from a human source. They're going to differentiate into other things. They will still have GFP, but can they also identify these human nuclei positive cells? So again, I'll show you this on the next few slides, but one of the things that they were able to see is that they were infiltrating not just in the area where they implanted, but they spread out through the entire forebrain, and they spread through both hippocampi as well as into the cortex. Okay. Any questions on this before we go on? No? So this is a really busy looking slide. So I'm going to take you through it. DAPI we've seen before. We use DAPI to stain nuclei, and it's really hard to see these individual little blue dots. Any cell which has DNA will stain for DAPI. GFP, it's not something that we normally find in the brain at all of any species, and we're seeing this green fluorescence protein here where we see these arrowheads. We can go back in and stain for human positive nuclei. So are the GFP cells also positive for human nuclei? They are. And we don't see the human nuclei anywhere where we don't see GFP. Perfect, because that's what we would expect. And are these actually forming normal resident cells? Does anyone remember what in the adult brain, in the cortex, we use GFAP to stain for? Actually, it's on the slide. So, sorry. So we can stain for the GFAP, glial fibrillary acidic protein, in the adult mouse forebrain for astrocytes. So these cells that we transplanted when they were really young, when we look at them in the adult, have formed astrocytes. We know that they're GFP positive, so that's good. We transfected them with GFP. They have human nuclei. We only see them in those green cells. And we also know that in this case, they have also formed these astrocytic cells. Okay, so we start to see them spreading out over time, and we start to see this stellate-like appearance, which you see here. The astrocytes are originally named astrocytes because they look like stars, and this stellate star-like appearance is very characteristic of different types of astrocytes. And at 5 months all the way through to 11 months, they would be able to track to see if they are EGFP positive, human nuclei positive, as well as GFAP positive. And they are. They spread throughout the corpus callosum. They spread throughout the different cortical layers, like cortical layer 5 as well as 6. And when they start looking, they see it spreading out through the entire forebrain. Do you have any questions about the technique in detecting the cells that they transplanted at all? Any questions about the staining or anything else that I can answer for you? No? Okay, so here's where things become a little bit interesting. I'm going to ask you for a little bit of help here, and you can tell me. So CA1, why is that of interest for us? It's part of the hippocampus, correct? But what else is interesting in this particular scenario? And tell me what DG is. So that's the dentate gyrus. So we know that we have dentate gyrus, CA3 over here, CA2, and then we have this CA1 layer over here. Perfect. That's the anatomy of the hippocampal formation. Um, and where do you see green, which represents green fluorescent protein? Yes. We see a lot around the dentate gyrus. Great. And we see a little bit up here in the CA1. So within the hippocampus, there were lots of these stellate-like green cells, the ones that we transplanted. We don't see GFP unless we transplant it through those pure populations of glial progenitors. And the highest density was actually found in the dentate region, okay? And you can actually see that here. This is the dentate gyrus. This is the dentate region. Okay, some of you have orange paper near your desk. Can you really quickly map out the connectivity and the different fibers that connect different areas of the hippocampus? Just do it real quick. Let's see if you guys remember, because it becomes important in a moment. So what are the fibers, for example, that innervate the CA1? What are the fibers that innervate, for example, the dentate gyrus? Do you remember all of those? And then show me if you guys have, you think you have it. Okay. Does anyone have a good diagram? No. Yes? No? No. Oh. So let me ask you this, and you can tell me if you recall this. What innervates or what sends projections into the hippocampus? Entorhinal cortex, great. And I don't need you to memorize those details. It's not on the slide. There is one, though, that I need you to actually tell me because I need to know if you know. Into the dentate gyrus where you see lots and lots of these glia. So if glia, and the hypothesis is the more glia that we have that are humanized, the better learning, memory, plasticity, everything else is going to be. Can we actually demonstrate LTP learning, like the electrophysiological basis in the dentate gyrus? How many of you think we can? Yes? Okay, one person. Anyone else agree with Danny? Danny's a pretty good bet, by the way. Like if Danny says yes, it's like 51% of the time he's correct. 51. Anyone else? Yeah, Richard. You definitely will. It's a very characteristic place where you find LTP. This is where we classically look for LTP, but my question is not, is this the better place? Can we see LTP down here in the dentate gyrus? CA3, we don't see NMDA receptor forms of LTP, but can we see LTP here, dentate gyrus? You guys are like lawyers. You're skirting around the question and kind of like, you know, this, that. Yes or no? Simple answer. Agree with Danny or disagree with Danny? Yes. Good job, Danny. Good job, class. So here, within the dentate gyrus, by stimulating the fibers that will make synaptic connections to the dentate gyrus, we can also observe LTP. It's not the preferred area because the entorhinal cortex actually sends in a number of projections into the dentate gyrus, and one of those projections is known as what? Which fiber pathway? No, not mossy fibers. Nope, that's the outflow. Yeah, it's the perforant pathway. And what you're going to see a little bit later on is that, yes, when we measure from cells in the dentate gyrus and the surrounding astroglial cells are really dense, the human astroglial cells are really dense around the dentate gyrus, can the LTP that we see in the cells of the dentate gyrus actually be enhanced? And so this becomes a really important point because normally we don't do or show you experiments that lead to the stimulation of the fibers that you can't see here, the perforant pathway fibers, but we can actually stimulate them. We can put in an electrode and stimulate those. Any questions before I go on? So one more thing. I told you at the outset, the glial cells of the human are different than the glial cells in the mouse. What this is actually showing you in this diagram in G, this is exactly what you see. This is a human glial cell. It is stained with GFAP, it has EGFP, has the human nuclei in it, and here, right beside it, is the is the mouse glia. So the mouse still has its own glia, but right beside it, dominating right beside it, is this human glia. And is there actually a difference? If you were to take a look, the human glia over in H are actually much, much bigger. So they have retained sort of the normal morphology. They're bigger cells than the normal mouse astroglia. Okay. So when you transplant these things in, do they stay human? Yes. And do they still retain the features of being bigger and more complex? I've shown you that they're bigger at this stage. I've shown you where they're localized around the dentate gyrus. So these human glial progenitor cells also stay and have the characteristic features that I showed you before. So they send out all of these massive processes. So they're bigger. They also make more elaborate connections than their mouse counterparts. They send projections, as normal astrocytes do in your brain, all the way to these blood vessels that are being shown to you here. So each of these end feet that you learn about in other courses are where the astrocytes are making connections directly onto the blood vessels. This is in the mouse brain. Mouse astrocytes normally don't make these elaborate types of connections. And these long processes were described as being torturous. They were making these end feet connections directly onto these blood vessel walls, and they looked a lot like the types of glial cells that we would see in our brains. We don't see these in mice, but we now see them when we put human cells into mice brains. So they have retained all of their unique human properties, and they are centered around the forebrain. They're going up into the dentate gyrus. They send long projections that we can measure using GFAP as well as EGFP, and they look like our astrocytes. So, and again, these long astrocytic processes, which span tremendous distances, like 600 microns might not sound like a lot, but for one process of one cell, that's a significant distance for this to be traversing. And you can see the normal mice astrocytes right beside it, small, not sending out these long processes at all. So these are the normal processes that you see in red. And you can see the one that is coming from the humanized astrocyte that has differentiated is much, much longer. And each of these sort of arrowheads represents one of the mouse astrocytes that we're still able to pick up. So do they still behave in other ways, functionally, not anatomically, not in terms of histology? Do they behave like they normally would? I told you one of the things that human astrocytes do is that they form a network. They form a syncytium, in other words, a connectivity, a network between each other. So these astrocytes, one of the things that we can do is inject in to these astrocytes a fluorescent marker, which is known as ROD2M. Richard, did you have a question? No? And when you take a look and you inject in to one of these cells, which is labeled with EGFP, so we know that the EGFP is only found in our progenitor cells. It's not found normally in the mouse brain. We've injected this into, we've injected the glial cell progenitors. It's now become an astrocyte, and this astrocyte we find under fluorescence, and then we take this astrocyte that we're seeing under the microscope, and then we inject this astrocyte here with ROD2M. Okay, so ROD2M is a fluorescent marker. It can only be spread if there are connections between cells. So if there are gap junctions, for example, between cells, it can spread from the cell that we injected it into its nearby neighbors. So one of the things that they found was if we take this EGFP positive cell and we inject this EGFP positive cell in the bottom panel with ROD2M, when we go back and we look at where is ROD2M now, if it was not connected, we would see zero staining except in the cell that we injected. But what they saw was the neighboring cells that were mouse glial cells, they're GFAP positive but not EGFP positive. One of the things that they saw was that the neighboring mouse glial cells also took up ROD2M. In other words, it suggested that they were all functionally coupled. There's no way for this dye to move from cell to cell unless they are functionally connected through gap junctions. And these functional gap junctions were formed between the mouse and the human glial cells. Okay. Any questions on this before we go on? So functionally, this is just like we see in our brains. They have a functional syncytium. Yes.
Did they also show that this wouldn't happen if you injected this into mouse glial cells that didn't have human glial cells?
So that's a great question. So if I took other mouse glial cells and I put them in, would they not form the syncytium? And I don't show you this data, but that's one of the things that they do show, that other mouse progenitor cells, when they're allowed to differentiate, don't actually form the syncytium. It's a great question, and I'm glad that you asked it. Okay, so why do these glial cells have this specialized role? So one of the functions that we think happens is that on these glial cells, they have all of those metabotropic neurotransmitter receptors, just like you would see on neurons, and they detect levels of glutamate as well as GABA. And they will also activate intracellular signaling pathways, including like elevating calcium. And they spread this signal out after the metabotropic receptors get activated through this cytosolic calcium to their nearby neighbors. And it, again, I'm not going to show you this data, but other papers have shown that if you can get a network of activity, it actually enhances synaptic plasticity, primarily through these glial cells and cytosolic calcium. So again, this is a really busy slide, and I don't need you to know all of it. Some of it, by the way, you should know, or you should be able to tell me a little bit about. Um, can anyone tell me what is going on in figure B? Just like in general terms. What are we looking at? Yes, at the back, Eddie. Okay, so you're right, 100%. So let me take you through A first. What this shows is that if you have humanized glia around it, for the same amount of stimulation, you get a much bigger response with human glia. So this is the slope of the EPSP, which means that for the same stimulus, like 0.06 milliamperes, if you have a chimeric mouse, the current is much, much bigger. And you can see that overall, the current that we generate is much, much bigger. But what this is showing you in B is that if this is normal LTP, and this is the response that you get with LTP, this increase after 100 hertz stimulation, and they actually did an insane protocol. This is not a normal protocol, and this is one of the things that hopefully as you become better neurobiologists, you'll learn. This is not a normal protocol. 100 hertz, yes, but they delivered 100 pulses at 100 hertz for 30 seconds. That's like not just tetanus or a high frequency stimulation. This is like uber tetanus. It's like not getting an electric shock when you like walk up to the chalkboard on a really humid day. This is like sticking your finger into an electric socket. This is not normal. This is an abnormal form of high frequency stimulation. But when they do this, they certainly see that there's an enhancement. This enhancement is known as LTP, and this LTP is even further enhanced if you have human glia in the neighboring regions. Now, they are doing this, by the way, at the level of the dentate gyrus. They are stimulating the perforant pathway. So they do have to increase the high frequency train a little bit, but this is really much more than you would expect to see, even for the dentate gyrus granule cell layer. Okay, so they are seeing an enhancement of LTP with this human glia. LTP, this enhancement, for the same stimulus, you get a much bigger response. This LTP suggests that these mice have enriched learning. Now, they haven't shown that yet, and I'm going to show you this in the next slides. And again, we won't go through all of the detail. Maybe I'll come back to it some other time, but this is a sort of a proof of concept. Um, one of the things that they're trying to show is, why does having human glia, sure, there's complexity, there is a syncytium. One of the other things that they try to show here is that human glia actually release TNF alpha. And this TNF alpha that gets released from these human glial cells actually enhances the GLUR1 mediated response. And we won't worry about the panel in A. And what this is showing you in B is that GLUR1 levels are enhanced, but NR1 levels are not. So AMPA receptors are elevated with the GLUR1, but the NMDA receptor levels stay the same, exactly as we would expect. Again, through this TNF alpha mechanism. So TNF alpha, when they apply it to a slice, also increases serine phosphorylation on the AMPA1 subunit at serine 831, but not at serine 845. Again, another strong biochemical indicator of LTP in this slice. So all of these are characteristic that in these humanized mice, that this LTP, this enhancement of LTP that they're seeing is actually because these humanized glial cells are releasing TNF alpha. And I don't know if you have ever encountered this before. It's not something that we use a lot of in neuroscience. There is a compound that some of you might have heard about in developmental biology called thalidomide. Have any of you heard of thalidomide? What is thalidomide traditionally in pharmacology, for example, been suggested to do? Yes. It used to be, yes. It used to be taken by women for morning sickness, and what was the result? So that was one of the big issues, and of course there were deformities with the children that were where there were deformities in the limbs of the children that they had. But one of the things that we use thalidomide for is that it can actually, in glial cells and neurons, wherever TNF is normally produced, it actually inhibits TNF alpha production. So when you apply thalidomide to the humanized glial cell culture, one of the things that happens is this enhancement that you normally see after you apply thalidomide, which blocks TNF alpha, goes all the way back down to normal levels. And that's one of the proofs that they have, that because they're using thalidomide, which blocks TNF alpha, that the enrichment that they saw here of LTP in panel E, and it disappears with thalidomide back to normal baseline levels, is actually because of increased TNF alpha production. Now, again, some of this, if you go through the literature, some of it is political. If you read through all of the references that you see at the back of this particular journal article, they cite Rob Malenka's group multiple times. Rob Malenka, who's a huge researcher down at Stanford University, as well as Dave Stellwagen, showed very clearly that TNF alpha enhances LTP. And I'm pretty sure he was a reviewer on this paper and asked that all these things were done. But again, whether or not that's a really good proof, I'll leave that up to you. It is, the data is what it is. It definitely shows that after thalidomide in gold down here, that the LTP that we normally see in red is decreased back to the levels that we would have without humanized glia. Any questions before I go on? Yes.
Were the LTPs measured in vivo or did you slice cultures?
Not slice cultures. These are ex vivo slices. Yes, Wenchen.
If thalidomide is what's contributing to the increase, if TNA alpha is what's contributing to the better LTP, why wouldn't they just inject TNA alpha instead of doing
So actually, one of the things that they show here, and I'm not going to take you through the proof, is you take a mouse, normal mouse cell, normal mouse hippocampus, you put in TNF alpha, the levels go up, okay? The LTP goes up, it behaves just like you would with humanized mice, and that's what it's showing you here in A. I don't know if it's a great proof or not. It could be multiple factors, and TNF could just be one of them. I'm not too sure. So again, I'm not going to go through this in a lot of detail because I know some of you haven't covered this in some of your neuroscience classes. But one of the things that you have to do, so yes, LTP is enhanced We think LTP is enhanced because of TNF alpha, because we can block it using thalidomide. It goes back to its normal levels. We think that the glial cells are what's causing that to occur. But one of the things that you have to demonstrate in every paper that you read is not just cellular physiology, not just LTP or electrophysiology, but you also have to demonstrate that there is a behavioral outcome. If these mice really do get smarter after injecting in human glial cells, they should behave like they're smarter. They should have memory like they're smarter. Since they're surrounded at the level of the dentate gyrus, we're not going to use the Morris water maze like we normally would with CA1. So there are a couple of things that they look at. And one model is this contextual fear conditioning. And this is freezing behavior. And this freezing behavior that they exhibit, where the chimeric mice, the one that have human glia in this middle panel, you can see that they freeze a lot more often. In other words, even after they've only learned it for one day, they're already freezing more often than their non-chimeric counterparts with just normal mouse glia. Does that make sense to all of you? So the contextual fear, do you like that data? Yes.
Why are the normal mice not?
Ah, good question. So these are normal mice down here. How come they're not learning at the same level? You're not going to expect it to be way up here, but you should expect that after day one, they've got some contextual fear learning, and it should continue to go up. Maybe not as quickly, but it should go up. Their mice, when they are comparing the red bars to the black bars, the control mice are not learning as fast. Again, don't really know. They don't try to explain that, but it is a good thing to take note of. Again, the other thing that they take a look at is another sort of hippocampal formation test, which is the Barnes maze. And then this was also shown to be disrupted or enhanced in the mice that had the human glia. The novel object recognition, which is sort of a learning independent model, also they demonstrated that in this particular case as well, the chimeric mice also behaved much better. They remembered having a new sort of toy put in the cage with them, and they spent more time with that toy in this novel object recognition. So by every measure that they looked at, the behavioral tests showed that for hippocampal and dentate gyrus dependent forms of learning, like contextual fear and the freezing behavior with contextual fear, and the Barnes maze and novel object recognition, all of them showed that a significant improvement in learning as well as in memory. So they retained that memory, especially in the novel object recognition. They could make, by the way, and they did this experiment where if you inject these animals with thalidomide, all of that improvement disappears as well. Okay, so maybe thalidomide is working through TNF alpha, don't really know. So all of this is really important because it shows that maybe astrocytes and human astrocytes, which are much more complex, actually lead to us being better, thinking better, having better memory, having faster learning. Again, it's not a definitive proof. Only one group has followed up, and it's their group that followed up 1 year later. No one else has really done these types of experiments. And they definitely showed that the human astrocytes in the mouse brain are bigger, are more complex, do make functional syncytia. They make a good case, not an excellent case, but a good case that this might be mediated by human TNF alpha causing increases in synaptic plasticity through the release of TNF alpha and enhancing that AMPA receptor current that we see in LTP. But we don't really know because it's really one group that's reporting all of this data right now until other groups are also coming out independently and showing exactly the same thing or using different tests showing exactly the same thing. Right now, it's a very interesting phenomenon that maybe our glia are really what make us all that smarter. Again, they didn't do these types of controls, which you would kind of want. You know, when these glia, before they start forming these complex conditions, before they start releasing lots of TNF, as a control, they probably should have done it at a much earlier age as well to show that there is this developmental increase. So I know some of you really hate those really long, lengthy announcements that I have on the announcements page. Apologies on that. I just recently heard about this TLDR wasn't really sure what that is until fairly recently. Some of you might find it useful. If you don't find those little summary things like no slide 23 or whatever from the thing, don't bother reading it. It's just going to be a long list of things. I am going to post them for this week. This is it in terms of what you need to know for your midterm. Next week, we're going to do the schizophrenia GWAS study, and we'll talk about schizophrenia, and we'll talk about the physiology and the neuroscience and genetics of schizophrenia, but you're not responsible for it. And Thursday, we're actually going to be doing a review session. Apologies on one last thing, while I have most of you here. The presentation dates, we are going to move to the Thursdays. For those of you who are wondering, we'll just have the Thursdays blocked off for this. I previously mentioned it as a Tuesday, and again, my apologies on that. Okay, any questions before we go? No? Yes.
Just a question. For the animal learning test that they did
Yeah.
they also tested it on the allograms as well?
They did.
Okay.
I didn't show all that data, David, but yes, they did.
So,
Yeah, no improvements.
okay.
Good question. Okay, have a great week. I'm around for office hours if you need to chat with me, and I'll be happy to do so.